Hello friends, we are hoping that you all are doing great and must be taking help from our videos. So, one more time we are here, presenting a new topic that is requested by one of our followers. In this video, we'll learn the heart is a pump and the rhythmical excitation of the heart. Let's begin. How heart functions as a pump. The human heart is an unusual and efficiently designed pump. Its main function is to pump blood through the entire circulation to meet the hematologic requirements of all cells of the body. This pump is most unusual in that the source of energy for the pump is located in the walls of the pump itself. It also has an unusual circulatory system, a system which must function in an organ, which shrinks and dilates many times a day as it pumps blood. The main vessels are located on the surface of the heart, and the smaller vessels, which deliver the blood to the cells of the pump, penetrate the wall, the source of power. These smaller vessels are so constructed that they can deliver blood to each cell even though the vessels are being firmly squeezed upon during systole, and must function in the organ while it is continuously changing in size and shape during the cardiac cycle. The major volume of blood flow occurs during diastole, when tissue pressure is lowest. Now, we will discuss rhythmical excitation of heart in detail. Rhythmical excitation of the heart is carried out by specialized excitatory and conducting system. The myocardial conducting cells that are 1% of the cardiac cells are the autorhythmic cells and form the conduction system of the heart. This diagram demonstrates the specialized excitatory and conductive system of the heart that controls cardiac contractions. It shows the sinus node, also called sinoatrial or SA node, in which the normal rhythmical impulses are generated. The internodal pathways that conduct impulses from the sinus node to the atrioventricular or AV node. The AV node, in which impulses from the atria are delayed before passing into the ventricles. The AV bundle, which conducts impulses from the atria into the ventricles, and the Purkinje system of the heart, including the left and right bundle branches of Purkinje fibers, which conduct the cardiac impulses to all parts of the ventricles. It also shows atrial internodal pathways and ventricular bundle branches. Now, we will learn each of them in succession. First is sinoatrial or SA node. It is a small, flattened, ellipsoid strip of specialized cardiac muscle, about 3 mm wide, 15 mm long, and 1 mm thick. It is located in the superior lateral wall of the right atrium, immediately below and slightly lateral to the opening of the superior vena cava. The fibers of this node are each only 3 to 5 micrometers in diameter, in contrast to a diameter of 10 to 15 micrometers for the surrounding atrial muscle fibers. Cells are connected through gap junctions. The cells of SA node at the top of the heart are known as P cells, that is, the pacemaker of the heart, because the rate at which these cells send out electrical signals determines the rate at which the entire heart beats, that is heart rate. It is supplied by the SA nodal branch of the right coronary artery. Next are interatrial pathways. They extend from SA node within right atrium to left atrium. They are comprised of small bundles of atrial fibers. The largest is anterior interatrial band. Velocity of conduction is 1 meter per second. It is supplied by the SA nodal artery. Following them are internodal pathways. They extend from SA node to AV node. They contain Purkinje type of fibers. They are connected to AV node through very small transitional fibers. 3 discrete internodal tracts have been reported, including anterior, middle, and posterior. The 3 bundles of atrial fibers include anterior internodal tract of Bachmann, middle internodal tract of Wenschebach, and posterior internodal tract of Torel. Adjacent are atrioventricular or AV node. It is located in posterior septal wall of right atrium, beneath endocardium, behind the tricuspid valve, adjacent to the opening of coronary sinus. It is the only point of electrical contact between atria and ventricles. It has slow conduction velocity. It is connected to bundle of His through penetrating portion of AV bundle. It is called the secondary pacemaker. It is supplied by AV nodal artery. To come are bundle of His. They are comprised of Purkinje fibers, which are even larger than ventricular fibers. They connect penetrating portion of AV bundle to ventricles. They are connected through gap junctions. Succeeding are right and left bundle branches. They are comprised of Purkinje fibers. The distal portion of AV bundle divides 5 to 15 mm towards apex into right and left branches. They are located beneath endocardium and progressively divides into smaller branches. They are responsible for generation of cardiac impulse. What is self-excitation, or pacemaker potential? Let's learn. Cardiac cells are capable of autorhythmicity, or self-excitation. The SA node normally sets the heart rate simply because its cells have the fastest intrinsic rate of firing, that is 70 to 80 beats per minute. The AV node has secondary pacemaker activity. When the SA node fails to control the cardiac rhythm due to depressed automaticity or impaired conduction, the AV node functions as pacemaker of the heart. The cells of AV node normally discharge at about 40 to 60 beats per minute. The cells of bundle of His are also capable of spontaneous firing at a slower rate of 15 to 40 beats per minute. We will discuss the pacemaker potential of heart in detail afterwards. You should keep in mind that SA node, being the fastest in generation of action potential, therefore acts as pacemaker of the heart. Cardiac impulse is the action potential generated in the cardiac pacemaker and transmitted to the rest of the heart. Now that you have covered the anatomy of the cardiac conduction system, ask yourself these important questions. How does the SA and AV nodes work? How do the impulses travel through the heart? The simple answer to both questions is an action potential. Action potentials generated by the SA node spread throughout the atria, depolarizing this tissue and causing atrial contraction. The impulse then travels into the ventricles via the atrioventricular node or AV node. Specialized conduction pathways that are bundle branches and Purkinje fibers within the ventricle rapidly conduct the wave of depolarization throughout the ventricles to elicit ventricular contraction. Therefore, normal cardiac rhythm is controlled by the pacemaker activity of the SA node. Sympathetic nervous system increases the rate of SA node activity while parasympathetic nervous system decreases the rate of SA node activity. What is the mechanism of sinus nodal rhythmicity? Let's learn. Cardiomyocytes have the special ability to stimulate themselves, that is, self-excitation or automaticity. The initiation of the action potential is dependent on ion channels that allow passage of ions into and out of the cells. In the case of cardiomyocytes, they have fast-acting sodium ion, slow sodium calcium ion, and slow, fast potassium ion channels, among other important channels, that maintain ionic equilibrium. This automaticity is particularly enhanced in P cells. They have a lower resting membrane potential than the surrounding cardiomyocytes and transition cells due to these factors. There is a high concentration of extracellular sodium outside the nodal fibers. A relatively large amount of sodium channels are already open. There is a passive diffusion of sodium into the P cells between heartbeats due to the leaky sodium channels. The passive influx of sodium causes a slow rise in the membrane potential of the cell, bringing it progressively closer to the generation threshold of an action potential. Therefore, cardiac P cells of the SA node are more readily depolarized than other cardiac cells. The SA node is also intimately connected with the surrounding heart muscles via the internodal and interatrial conduction pathways. Consequently, the generated action potential can be rapidly transmitted to other cells. This enables the SA node to set the pace at which the heart cells will depolarize and subsequently contract, making it the pacemaker of the heart. On average, the SA node can fire between 60 to 100 beats per minute at rest. This is the action potential graph of heart muscle. It shows refractory period in which the cell is depolarized, that is, made more positive, plateau when there is little to no change, then repolarized, that is, made more negative. In absolute refractory period, ion channels are temporarily inactivated. In relative refractory period, some ion channels may be activated. Next we will discuss atrioventricular node and delay of impulse conduction from the atria to the ventricles. Although the main role of the AV node is to facilitate passage of the depolarization wave to the ventricles, it also has additional functions. In the absence of a functioning SA node, the AV node has the ability to take over as the pacemaker of the heart. Recall that it also has P cells that are able to establish an albeit slower rhythm, 40 to 60 beats per minute. The AV node is also responsible for slowing down the passage of the electrical impulse traveling to the ventricles. This important phenomenon allows more time for the ventricles to remain quiescent and fill with blood coming from the contracting atria. But how does the AV node slow down conduction? One key feature of the transition cells and P cells in the AV node is that they have fewer gap junctions at the intercalated discs. Consequently, there is more resistance to conduction in this part of the conduction pathway than there is in other areas. Now let's put all that information together and outline the cardiac conduction steps. The SA node generates the action potential. The action potential passes along the internodal and interatrial conduction pathways, causing atrial systole. The impulse arrives at the AV node and is slowed down to facilitate ventricular filling, that is, ventricular diastole. The impulse then passes from the AV node to the AV bundle. It is then rapidly dispersed through the bundle branches and subendocardial tissue, causing ventricular systole. What is cardiac plexus? The entire cardiac conduction system is under the influence of the autonomic pathway. Sympathetic stimulation of the conductive tissue comes from the cardiac plexus, while parasympathetic influence arises from the vagus nerve. Activation of the sympathetic nervous system results in the release of adrenaline or epinephrine and other adrenergic neurochemicals. They bind to beta-1 and beta-2 receptors, found in both the SA and AV nodes, as well as along the supporting conduction pathways. The overall impact of the sympathetic system is an increase in the depolarization rate of the SA node. Consequently, this elevates the overall heart rate, that is, increased chronotropy. Since these adrenergic receptors are also present on cardiomyocytes, the sympathetic drive will act on these cells, increasing the contraction force, that is, increased inotropy. Therefore, the overall cardiac output will be increased. On the other hand, the parasympathetic activation of muscarinic receptors at the SA and AV nodes will have the opposite effect when compared to the sympathetic system. Parasympathetic stimulation slows down SA node activation, thus reducing the heart rate. It also reduces the contractility of the cardiomyocytes, effectively reducing the cardiac output.